Yes, everyone, switch on your videos. Yes, everyone, switch on your videos quickly, quickly. Students, first of all, you people tell me one very important thing, that have you people started to do your self-study sessions? Have you, like, how much time do you people take out in a day for your self-study, apart from tuitions, apart from schools and everything? How much time do you take out for studying alone, like you by yourself? The question is to every student. Tell me, anyone can start. It's okay. It'll be different for everyone. So tell me. Tell me in terms of hours. I hope it's hours. I hope it's not minutes. Is it minutes? Or is it seconds? I hope it's not seconds, students. So tell me, how much time do you people dedicate to your self-study sessions?
Ma'am, because of school, it's less, but now we have homework and also for study it's 2 hours, or if I fall asleep, then 1 hour.
Okay. So basically 2 hours for everyone, right? 2.5 hours or 1.5. I don't think it'll be less than 1.5, because the syllabus that you people have is a lot, and it's not easy to cover every single thing in just 1 hour, isn't it? Now, the point or the reason why I'm asking you all this particular thing is that, see, students, it doesn't matter how good of a coaching you attend, it doesn't matter how good of a school you attend, unless and until you people indulge yourself in self studying, nothing is going to help you. Nothing. The best teacher that you people can have is, first of all, yourself. Getting my point? So what I mean to say by this is that, like, we are on like biomolecules chapter, right? We have completed so many chapters together. Now I want you people to be so prepared that no matter if it's me or if it's anyone else, if anyone decides to take your test, you will like, okay, I'm ready. I'm ready with my chapters. I want you all to be like this. I know it's very hard to do so. And I know the backlogs have been starting to pile up. Do you people have backlogs of chapters? Is it big, big log, or is it a tiny log? Tiny. Tiny for all? No. Big. See, the only thing that you people can do, I've been telling you this thing from day one, that once you're done with the chapter, read it, learn it, set it aside. Because once you sit before your exams with that pile up of a chapter list, you're going to get panicked, you're going to get worried, and then you'll be like, out of panic, you can't study more. So we have to avoid all of those things. You're getting my point? So the reason why I'm saying this is because your exams are approaching. Now, I have given my 100% to teach you all. I hope that you people feel that way also, that ma'am has given her 100%. Now, I want 100% from your side. Getting my point? Isn't it fair to expect 100% from your side? Is it fair or
Yes.
unfair? Lighten up. What is, I'm not scolding you. I'm talking to you all. Why are you people making such faces? And everyone switch on your videos. So the only thing, I don't want to tense you people out, but the only thing I want to say is that even though you still have a backlog, it doesn't matter. It's like right now also you can start doing your chapters one by one, and you have that liberty. You can ask me anything. It doesn't matter if we are doing biomolecules now and you have questions from morphology, or you have questions from biological classification. It doesn't matter to me. The only thing that matters to me is that you are studying, you are scoring well. Getting my point? So you people can start doing your chapters that are in the backlog also. Complete it. do the questions, make the questions, and just be ready for your test. Your tests are like in this month only, right? September, for all of you, most of you, right? Yes, so you need, when will you prepare for your test? One day before. It's not going to be possible to do it one day before. So from right now only you start preparing, okay? And while you are in the class, while I'm teaching you the topic, that time try to learn, like listen in such a way that you learn here only. Some traces of it would be in your mind still, right? All right. Now, in the previous class, we had discussed about the various kinds of sugars, right? We talked about the most simple sugars such as monosaccharides. Then we moved one level up and we talked about oligosaccharides, derived monosaccharides. And then ultimately we talked about the polysaccharide. That is the biomacromolecule. Right, now we are going on to the next biomolecule, that is the amino acids. Have you people ever heard about amino acids before? Like you
Yes,
might have
ma'am.
also, yeah, where have you heard?
Digestion process. It's the
Digestion. Yeah, it's there also. And you might have heard that, okay, eat this, this has all the amino acids, eats this. Like people say that eat particular stuff that contains a lot of amino acids. Now, why are amino acids so important to us? Remember one thing, that amino acids are the building blocks of proteins. In our body, we require protein, right? There are so many different kinds of proteins in our body. There are like contractile proteins, there are pigments in our body, enzymes are there, digestive enzymes are there, hormones are there. All of these things are proteinaceous structures. So when I talk about protein, we must also talk about what it is made up of. So every single protein molecule is made up of variety of amino acids. Okay, amino acids, just imagine them as being the building blocks of protein. So amino acids, they are the building blocks of proteins. Meaning these are the ones because of which proteins are made. Now talking about amino acid, so these are the organic compounds that contain, you people know functional group, students? All of you know
Yes, ma'am.
functional groups, right? Okay.
Yes, ma'am.
So there are variety of functional groups, out of which the amino acid has the amine group, which is NH2. It has the carboxylic group, which is COOH. And along with that, it has a variable group. So if you see the basic structure of the amino acid, this is what the structure looks like. In the middle, there will be a parent carbon, this one. Now this, you can say position of where the structural, or sorry, the functional group is present. It's not significant. Even if you rotate it like that, then also it will mean the same thing, isn't it? So when we talk about the carbon, how many bonds does a carbon make?
Four.
Hmm?
Three.
4. See, look at the structure. It makes 4 bonds, right? It is making a bond with COOH. It's making a bond with NH3, H, and here R. You see that it is making 4 bonds in totality, isn't it? So always remember the valency of carbon is 4. We do know that. And it makes 4 bonds with different molecules. Now, when we talk about amino acid, this is the basic structure of amino acid. This is how an amino acid looks like. Now, what did I say? One amino acid will have an amine group, which is NH3 or NH2. Then it will have a carboxylic group, that is COOH. It will have one hydrogen and it will have a variable group. This is not any functional group. Have you studied any functional group called as R?
No, ma'am.
No, there is no such functional group called as R, but R is here to denote denote the fact that it is a variable group. Variable means it changes. It changes from amino acid to amino acid. Clear, students?
Yes, ma'am.
So where did you all go? One by one, like one student switched off, second student switched off. Why? Why are you people disappearing on me? Everyone switch on your videos or else I will not teach.
Ma'am,
Hmm?
the amine group can also be written as H2N.
Yes,
Well,
it can. Not actually H2N, but see, I'll tell you. Abir and Sabiha, you also switch on your videos?
Yes, ma'am.
Hmm. See, the reason why we write the N before the H, because the carbon here is forming a bond with the nitrogen. Okay, and then nitrogen has a bond with hydrogen. So if you write like this, or if you write like this, usually, but try to write it like this only, NH3, because that is the functional group. So this is the basic structure of amino acid, and whenever you're writing in your exams, try to write the basic structure. Clear? Okay, Jaslyn. All right. So this is what the introductory part for amino acid is like. These are the organic compounds. And the best thing about it is that all of the functional groups that are present in the amino acid, each of them have their own particular function to perform. Each of them give a particular property or a characteristic to the amino acid. Now, moving forward, based on the nature of the variable group, meaning what is the kind of R, the R which is the fourth group that is attached, depending upon that, there are so many different kinds of amino acids present. In totality, students, there are 20 amino acids. That's it. I think, in the research currently, two more amino acids have been added. So all in all, 22 are there, but you people write 20. Okay. There are in totality 20 amino acids. And as I told you that they are the building blocks of proteins. Now, there are so many variety of, you can say different kinds of classification of amino acids. Amino acids have been classified in variety of ways. The first one is essential amino acid. You people understand the meaning of essential? What is the meaning of essential? Normal English meaning.
Something very
it's
necessary.
very
Yes, something that is important, something that is necessary. Now, see, they have given a very weird definition as to what will essential amino acids be. Now, you would think that these amino acids, since they are essential, so they will be present in our body, isn't it? We'll normally think that, but that's not the case. They say that essential amino acids are those amino acids which we cannot synthesize on our own. Our bodies cannot make these amino acids. So what do we have to do? We have to take them through the food, right? We have to eat them, like we have to take it through the diet. So the examples of the amino acid that are essential are lysine, methionine, phenylalanine, tryptophan, valine, isoleucine, leucine, and threonine. These are the names of the amino acid which are essential. Essential means they have to be taken through the diet, through the food, because our body cannot synthesize it. And apart from that, it is very important for us. It is indeed very important for us. Okay. Now, see, here in this particular part, which is amino acid, to learn the names of the amino acids, you have to make your own kind of mnemonics, because that is how you will be able to learn, or else not possible. Okay. So you can make your own, or I'll give you mine. When I was studying this particular topic, I used to learn it like LMPTVIL. Just learn LMPTVILT. So L stands for lysine, M is for methionine, P is for phenylalanine, T is for tryptophan, V is for valine, I for isoleucine, L for leucine, and T for threonine. So you have to make, you can use mine also, you can use yours also, but you just have to make mnemonics so that you don't forget it. Now see, there are many amino acids that starts with P, like phenylalanine is there. Apart from phenylalanine, there is proline also. So you have to pay attention to those technicalities also. Clear, students? Any doubts up until now? Okay. So this was about essential amino acid. Now there are some amino acids that our body can synthesize, but it can synthesize it slowly. Slowly the synthesis will take place. So those the example, like those amino acids that are synthesized by our bodies very slowly, so we'll call them as semi-essential amino acid. Semi-essential amino acid. The examples are arginine and histidine. Arginine and histidine. Lastly, there comes the non-essential. Now, a person would think that these are the amino acids that we do not need. But no, that's not the case. These are the amino acids that we do not need through the diet. We have it synthesized in our own body. Our body can synthesize it. So they are termed as non-essential amino acids. Now, the example for non-essential amino acids are alanine, cysteine, glutamate, glycine, and proline. See, this is the one, another P. Okay, so I made this ACG square P to remember it. You can make yours, you can use mine. It's up to you. This much is clear that three different kinds of amino acid based on their essentiality or where we obtain them from. Any doubts? Anything you want to ask me? All right, very good. Okay, now see. Can you tell me what is like, this is structure number 1, 2, and 3. Can you tell me what is the difference in between these three? Like, what is that one thing that is separating them all?
The side group,
Or
the
Yes, they have different R groups. Do you see that their basic structure is same? They have COOH, which is carboxylic group. They have amino group, and they have the hydrogen. But one thing they have in extra, it is the variable R group, right? Their R group varies from everyone. So glycine is that amino acid that has hydrogen as the variable group. And since it has hydrogen, which is not a functional group, it's just a molecule. So glycine is the simplest amino acid in terms of structure, in terms of chemical properties. It is the simplest amino acid. But when you look at alanine, alanine has methane, right? CH3. Here is hydrogen, here is methane. So alanine is the next. Then when it's attached with an alcohol, which kind of alcohol will it be, students? Have you studied nomenclature?
methanol.
Methanol, correct, because meth is for one carbon, isn't it? So when methanol is attached onto this basic structure of amino acid, we'll call it as serine. When methane is attached, we'll call it as alanine. And when hydrogen is attached, we'll call it as glycine. Clear? They have given just the three basic ones, the very simpler ones. There are many amino acids, like imagine structures for 20 amino acids that you don't have to remember, but at least remember these three. Zarab, your question says amino acid said it's not a homopolymer. See, it's not about amino acid. Amino acids are micromolecules. Okay, biomicromolecules. They are not macromolecules. Okay. When you talk about protein, which is made by the amino acid, the proteins are heteropolymers, meaning they are made up of different kinds of amino acids. Getting my point? And when you talk about amino acids, they are the most simplest structures. They are biomicromolecules. They are not macromolecules. Okay. So if anything is a biomicromolecule, then that means that it is not made up of monomers, right? It is itself a tiny moiety. Now, furthermore, the amino acids can also be acidic, basic, and neutral, depending upon the charge. Which, when I talk about acidic charge or anything that is acidic, which charge contributes to acidic nature, positive or negative charge?
positive.
Hmm? Positive, negative. When the negative charges are there, we'll call them as acidic area. Okay, that will become acidic. And like just like how if we talk about the basic part, right? Basic means when more and more plus charges will be there, we'll call that as basic. When more and more negative charges will be there, we'll call that as acidic. And when the number of positive charge and the negative charge will be equal, we'll call it as neutral. So acidic amino acid, these are those amino acids that have an additional carboxylic group. Now, have you people studied this, that if we talk about COOH, if we remove this hydrogen, the COO gets a negative charge. Have you studied this? And when we talk about NH3, if you remove one H, it'll get a positive charge. Okay. Meaning if you remove electrons from any, not remove, if you add electrons, it will get the opposite charge of it. Anyway, but the thing that I wanted to tell you people is that normally this, if you can see this structure, here carbon is present, hydrogen is present, carboxylic acid is present, and the amino group is present. But in addition to the carboxylic group that has already been there, it will all like again also have one more carboxylic group, COOH. So that is the case of acidic amino acid, because it will have two negative charges now. Okay, one on the other COOH, one on this one. So depending upon the charge, the negative charge, the acidic amino acid, they are glutamate and aspartate. Glutamate can also be called as glutamic acid. Aspartate can also be called as aspartic acid. So these are the ones that have additional carboxylic group present in. Clear? I'll write it here. That whenever anything is acidic, it has more negative charge. Clear? Now after that comes the basic amino acid. Now basic amino acid is that amino acid that have additional amino group. It will have additional amino group. So remember that basic is that condition when they'll have more positive charge. The example for the basic amino acid are lysine and arginine. And this is the structure of lysine. If you can see, this is the normal parent molecule amino group, but it has an additional amino group as the R group. So we'll call it as the basic amino acid because now it will carry positive charge, two positive charges. Clear? And when the amino acid has equal number of acidic and basic character, meaning positive and negative charge are equal, we'll call it as the neutral amino acid, such as valine, alanine, glycine, leucine, and isoleucine. Okay, and I used this word to remember it, vaguely. Like how ugly is there? So vaguely, to remember them. Like once you learn the different names, then the mnemonics will be easier. Any doubts up until now?
No, ma'am.
Okay. Now there are some amino acids that have a sulfur group present in it. Sulfur, S. So those are cysteine and methionine. Also cysteine. Okay, cysteine, methionine, and cysteine. These are those amino acids that have additional sulfur group. And lastly, there comes aromatic amino acids. Have you seen these cyclic structures in chemistry? What is the structure for?
Benzene.
Benzene. Very good. So you people know what are aromatic agents? Anything that has an
Smell.
aromatic ring, isn't it? They have the aromatic ring and they are aromatic because they have some sort of aroma, sweet fragrance. Benzene is a sweet smelling component. So those kind of amino acids that instead of the like in the place of the variable group, they have the aromatic ring. Just like how benzene ring is there, several other rings are also there. So that is the case of aromatic amino acid. It is the example is tyrosine, phenylalanine, and tryptophan. Any doubts up until now? No? Okay. Now talking about the special properties of amino acid, but first you note down till here.
Ma'am,
Yes?
ma'am, in the bonding there is a R, right, which is known as a variable group. It is also called as alkyl group because in the reader it's written like that.
It is also called as which group?
Alkyl group.
Spelling?
ALTYL.
Altyl. Yeah, it can be another name for it. I don't know this, but it is variable only. Okay, variable or extra group. Okay, let me check. You note down, I'll check it once.
Yes.
There it's written alkyl, ALKYL, alkyl.
Yes, ma'am.
Okay, okay. Yeah, it can have alkyl group also in that. Alkyl groups are what? These are the groups that do not have just one hydrogen. They miss one hydrogen. Meaning if you remove one hydrogen from alkane, it will become alkyl. I thought you were saying altyl. I did not know what that thing was. Alkyl group, yes, alkyl group can be present. Alcohol can be present. Then carboxylic, or any group can come and attach onto the variable group. And
Ma'am,
that
I
will define.
alkyl only.
Hmm?
I meant alkyl only, not alkyl.
Altyl.
Yes, ma'am.
I don't know this then. I don't think there is anything such as altyl.
Because in the reader it was given like that.
Okay.
Done, ma'am.
All of you are done?
Yes, ma'am.
Done, ma'am.
All of you are done, students?
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
No, ma'am.
Okay, okay. Take your time. It's all right.
Done, ma'am.
Okay, everyone's done?
Yes.
Yeah. Done, ma'am. Done, ma'am.
Everyone's done?
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
All of you are done, students?
Yes, ma'am.
Done, ma'am. Done, ma'am. Done, ma'am.
Everyone's done?
Done, ma'am.
Everyone's done?
Yes, ma'am.
All right. Now talking about a very special property that the amino acids have is that they can exist as a very special structure which is called as a Zwitter ion. Meaning, see, this you can tell by the functional group that the amino acids, they are ionizable in nature, meaning they can either have a positive charge or negative charge. But there comes a condition wherein amino acid exists in the form wherein it has both the charges, and that is a stable form, sort of like this one. See, A is the condition of a positively charged, and B is the condition of a C is the condition of a negatively charged. But if you look at B, this one molecule has positive charge also and negative charge also. So this special property which is shown by the amino acid, this is referred to as the Zwitter ionic property. The molecule that has both the charges and it's not neutral. It's not cancelling out each other. So this is known as a Zwitter ion. So with this, we have completed the amino acid. Coming on to the next, that is protein. Now the protein, you know that they are the part of the biomacromolecule, right? And they are made up of peptide bonds, meaning several amino acids, that are 20 amino acids, they come together and make the proteins by the peptide. Now they say that the protein is a heteropolymer of amino acid. Heteropolymer means one protein can be made up of different types of amino acid. It's not necessary that it will be made up of just one type of amino acid. It is made up of different types of amino acid. Now, talking about the some of the examples, such as collagen. Now, see, collagen is the most abundant protein in the animal world because it is present in the skin, right? It is a protein. Collagen is a protein and it is very abundant in the animal world. And it is the main component of connective tissue of animals also. Collagen helps in the connective tissue also. And then there is another compound called as the riboscope, that stands for ribulose bisphosphate carboxylase oxygenase. This is the molecule that is required during photosynthesis. And this one is the most abundant protein in the whole entire biosphere. It is the abundant protein in the whole entire biosphere. Now, the proteins, they carry out multiple functions. See, proteins are very important for us because most of our structures in our body and the cellular structures also, many of them, they are made up of proteins. So let us see what are the functions that are performed by the protein. So the first function is the transportation of nutrients across cell membrane. So they act like many proteins, they act like transport molecules, and they can help in the transportation of nutrients across the cell membrane. Clear? That is the first function that it performs. Then the next is it fights infectious agents, meaning antigen antibody reaction. You people know what antibodies are? Antibodies are the molecules that are made in response to any foreign particle or any pathogen when it enters our body. So the protein, it fights the infectious agents. Now, some proteins can act as hormones, and almost, you can say, almost all of the enzymes are proteinaceous in nature. Now, talking about the protein and their functions. See, collagen, it is the ground substance found in the skin, found in the connective tissue. Trypsin is the enzyme that is found in the digestive system for digestion. Insulin is the hormone which is responsible for the glucose management in the blood. Antibody, these are the protein molecules that fight infectious agents, that is antigen. It fights the antigens. Then there are receptors. Sensory reception like smell, taste, hormone, all of these receptors that are there in the body, they are also made up of protein. And lastly, there is GLUT4 that is responsible for the transportation of glucose into the variety of cells in the body. So these are the proteins along with the functions that they perform. Any doubts up until now, students?
No, ma'am.
All right, now you people can note down. Then we'll move forward.
Done, ma'am.
Everyone's done?
Oh, ma'am.
Done. Done. Ma'am, could you give the definition for heteropolymers again?
Yes, heteropolymers are those compounds that are made up of different monomers. Compounds made up of okay. Done, students?
Done, ma'am.
Done till here?
Yes.
Okay. Any doubts up until now?
No, ma'am.
All right. Now see, in this we have seen two examples. One is the collagen, which is the most abundant protein in the animal world, right? And then the ribulose bisphosphate. So these two things are clear to you all?
Yes.
Okay. And you've understood the heteropolymers also?
Yes, ma'am.
Okay, okay. Very good.
Done.
Everyone's done, students?
No, ma'am.
Okay.
Done, ma'am.
Okay, everyone's done? Done, students?
Yes, ma'am.
Done, ma'am.
Okay, everyone's done till here?
Yes, ma'am.
Yes, ma'am.
All right. So this was sort of like an introduction for the proteins. Now see, the proteins, they also have a variety of structures that are associated with them. And all in all, proteins have four structures. Okay, there is primary, secondary, tertiary, and quaternary. Four structures of proteins are there, each of which have their own, you can say, unique properties. Example, and the use, like where they are used, what significance they carry, that is also what we are going to study under the topic, that is structure of proteins. Now when we talk about the primary structure of the protein, primary, see, many a times if I write primary or if I write 1 degree, it's the same thing. Okay, so primary structure is a linear structure of protein. It is a linear structure. You people understand the meaning of linear? Linear means what? A line like, right? A rod like, meaning a straight structure is there. Now, this is the sequence in which the amino acid in a polypeptide chain are present in a very linear, or you can say one by one manner. That is the primary structure of it. Now, when we talk about the primary structure, so it gives information about the first, second, third, etc. position of the amino acid. Meaning, By this, we mean that which amino acid is first in position, which amino acid is second in position, and which one is third in position. That is what you can understand when you talk about a primary structure, or when you look at the primary structure of a protein. Now, in this case, like, since it is a linear structure, it is a line-like structure. So the first amino acid will make the N terminal of the amino group, meaning it will be present at that side by the amino group. And then the last amino acid will give the C terminal because it will be present from the carboxylic acid group. So this is about the primary structure of the protein. Then there will come the secondary structure, tertiary and quaternary. But all in all, you people have to understand that each of these structures, they have some kind of stability, some kind of function that they have to perform, and their significance is 100% there. Okay? Any doubts up until now?
No, ma'am.
All right. Okay, you can note this down. Then a polymer is not a thing. I mean, it is a name of a thing. For example, if you talk about protein, it's a polymer. If you talk about nucleic acid, that is also a polymer. So polymer, they are bigger structures that are made up of smaller structures. So remember that the polymers, they are made up of monomers. And since many smaller units, they come along and they get attached, they form a poly. Poly means many. Mer means unit. Clear?
Yes, ma'am.
Okay, everyone's done till here?
Yes,
No.
ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am.
All right. Okay, students. So we'll keep it till here only for today's class. Now, see, all of you can exit the class, but Jesslyn, please stay. Okay? The rest of you can leave the meeting. We'll continue from the secondary structure in the next class. Okay?
Yes, ma'am. Thank you, ma'am.
Thank
All
you,
right.
ma'am.
Okay. Good night, everyone. Okay, am I audible, Jesslyn?
Yes, ma'am.
All right. Please switch on your video. Are you facing some internet issues today?
Ma'am, my laptop is lagging a lot.
Yeah, I can see that. Okay. See, I'll be asking you some questions and you'd have to answer them to me, okay?
I
You
didn't
like,
study for Plant Kingdom.
hmm?
Ma'am, I didn't study for Plant Kingdom. They
But you
didn't.
have given the test, right? For Plant Kingdom. If you have given the test, then you have already studied, isn't it? It's like basic questions. It's not a lot. It's not very tough question. Some tiny mindy, because like I want to track the progress of my students. So that is why I will be asking you some questions and you'll have to answer. Okay. Now,
Okay, ma'am.
yes. Now, when we talk about the plant kingdom, we studied about the systems of classification, right? So, what are the different kinds of systems of classification?
Sorry, ma'am.
What are the different kinds of systems of classification?